Cells Part 2 lecture. The objectives of this lecture are to compare and discuss RNA and DNA and their functions in protein synthesis, and then also discuss the stages of meiotic cell division. DNA and RNA play a crucial role in protein synthesis and mitosis. DNA is a molecule with genetic information. DNA molecules resemble a long, narrow ladder twisted around and around its axis and is shaped like a double helix. Each molecule is made up of sugar, bases, and phosphate units. Bases are nitrogen-containing chemicals, and they are adenine, thymine, guanine, and cytosine. Each body has 46 chromosomes. The function of genes. Genes are copied from DNA and are copied to RNA in the process called transcription. RNA transcripts are used to process in a process called translation, in which the code that determines the sequence of amino acids is translated to form a protein. The structure determines the role of the protein, whether it is the structure or in function of the body. DNA has complementary base pairings. Each step of the DNA ladder contains a base pair. Adenine and thymine are paired, or cytosine and guanine are paired. A gene is a specific segment of base pairs in a chromosome, and the genetic code is a sequence of base pairs which determines your heredity. DNA is contained in the cell's nucleus, and protein synthesis occurs in the cytoplasm. Thus, genetic information must pass from the nucleus to the cytoplasm. The process of transferring genetic information from the nucleus to the cytoplasm where proteins are produced requires completion of transcription and translation. Here's an image that shows the whole protein synthesis, which begins with transcription of mRNA, which forms along one gene sequence of DNA molecule within the cell nucleus. mRNA transcript leaves the nucleus through the nucleus pores. Ribosome subunits attach to and begin at the beginning of the mRNA molecule and begin translation. In translation, transfer of RNA or tRNA molecules bring specific amino acids and are encoded by each mRNA codon and puts them into place at the ribosome site. As amino acids are brought to the proper sequence, they join by peptide bonds to form long strands called polypeptides, which is number 4 here. Several polypeptide chains may be needed to complete the whole protein molecule. Transcription is a double-stranded DNA that is separated to form messenger RNA, mRNA. Each strand of mRNA duplicates a particular gene, base pair sequence, from a segment of the DNA. mRNA molecules pass from the nucleus to the cytoplasm, where they direct protein synthesis in ribosomes, or the ER. Translation involves synthesis of proteins in cytoplasm by ribosomes and require the use of information contained in mRNA. Codon is a series of 3 nucleotide bases that act on a code for a specific amino acid. Cells, genetics, and diseases. Abnormal DNA that is inherited or that results from damage is often the base of disease. Factors that cause damage to the DNA molecule include chemical or mechanical irritants, radiation, bacteria, and viruses. Many diseases have a cellular base, meaning that basically the cell problem, it's a cellular problem, but it may infect the entire body. Cell division. Reproduction of cells involving division of the nucleus, which is called mitosis, and the cytoplasm. Two daughter cells result of the division. A period when the cell is not actively dividing is called interphase. DNA replication is the process by which each half of the DNA molecule becomes a whole molecule identical to the original DNA molecule, which precedes mitosis. There are 4 stages of cell division: prophase, metaphase, anaphase, and telophase. Mitosis is the process of cell division that distributes identical chromosomes or DNA molecules to each new cell formed when the original cell divides. It enables cells to reproduce their own kind, which makes heredity possible. So here's a picture of the phases of mitosis. So interphase is where the cell is doing nothing. Prophase is the very first stage. Then we move to metaphase, anaphase, and then telophase, which is the final stage before the cell splits in two. Prophase is where the chromatin granules become organized. Chromosomes, pairs of linked chromatins appear, centrioles move away from the nucleus, and a nuclear envelope disappears, freeing genetic material. The spindle fibers appear and are guide wires to assist chromosomes to move towards the opposite ends of the cell later in mitosis. The second phase is metaphase, which chromosomes align across the center of the cell. Spindle fibers attach themselves to each chromatin. Centrioles have migrated to the opposite ends of the cell at this point. Centromeres break apart. Separated chromatids, now called chromosomes. Chromosomes are pulled to opposite ends of the cell, and a cleavage furrow develops at the end of the anaphase. The telophase is the 4th stage. Cell division is complete. Nuclei appear in daughter cells. The nuclear envelope and the nuclei appear. Cytoplasm is divided, and the daughter cells become fully functional. Changes in cell growth. Anaplasia is a condition in which cells change in orientation to each other and fail to mature normally or differentiate in a specialized cell type. This is when the 4 stages of cell division are interrupted. Changes in cell growth. We have hypertrophy and atrophy. Hypertrophy is an increase in the size of the individual cells, increasing size of tissue. Atrophy is the decrease in size of individual cells, decreasing the size of the tissue. Hyperplasia is the increase of cell reproduction, increasing the size of the tissue, so there are more cells in this row than normal. Dysplasia is where there is an abnormal orientation and stacking of cells. Anaplasia is the production of abnormal, undifferentiated cells. Cancer is pictured here, so this is below here is a little excerpt from the lung, and what you can see here is an abnormal mass of proliferating cells here in the lining of the lungs, And then as you can see below, the cells are now entering the lymph and blood systems. So this would be considered a malignant tumor.